That's a great start. So welcome to Sharon's Defense. This is, of course, a joint effort between Julie Baker, raise your hand, and me, and we're most appreciative, Julie. So today we're here to hear Sharon defend her thesis. Her thesis is entitled Characterizing Human Cellular Reprogramming and Exon Activation Through Single-Cell Gene Expression Analysis. Can everyone hear me okay? Is it even the back? The sound needs to be louder? Okay. I thought I would begin with representative images of Sharon's early development. So what you see on this slide, beginning on day 0, is the immature oocyte that was probably not completely programmed yet to begin Sharon. On day 1, you see a fertilized egg. The two pronuclei migrate towards each other, and as they're migrating towards each other, they're going through this extensive process of reprogramming of the epigenetic information. The day 1 embryo is obviously called a zygote. On day 2, you see the first cleavage division. Later on day 2, you see the second cleavage division. And then on day 3, the embryonic genome activates. Now what you see on this panel of figures is a perfect set of embryos. And so the assumption is that representative embryos of images of Sharon's early development would encompass absolutely perfect embryos. Perfect blastomeres that go on day 4 to form a perfect morula. On day 5, you can see the two beautiful cell layers with an extensive contribution to the inner cell mass at the whiter arrow, and then of course she hatches on day 6 and attaches. And what comes about through a series of developmental steps is shown on this slide. Ultimately, there is a connection between the first chapter of the book and the chapter that we're on now. And so you see some pictures of her with Blake. I'm not sure exactly why she's so either sad or mad in the middle of the upper hand of the middle there, upper middle. In the upper right, you see her taking on the role of a Roman goddess. A little bit to the left here, we see her on a lake drinking champagne, and then looking out curiously in the middle of the bottom panel. On the very right, she's mocking me with her glasses. And so it is just a great pleasure to have seen her development. Obviously, I did not see it from the beginning, but I have seen it through these stages here. And so with that, I'd like to do what has been traditional in my laboratory, and that is to run through the lineage. So I believe that we are all related in the scientific family, and so here is Sharon's academic family tree. I've traced it back to about the 1600s. And I'll try to be relatively rapid here. So at the bottom, what you see is Sharon, a picture of her in Alaska, and I am her academic mother. She's actually got 2 academic mothers because Julie stepped in. And my career, my doctoral degree was done in the laboratory of Tim Huffaker at Cornell University, and we studied meiosis in yeast. I also did postdoctoral work in the laboratory of David Page at the Whitehead Institute, where we studied human genetics and repetitive sequences in the Y chromosome. Both Tim and David did postdoctoral work in the laboratory of David Botstein. Sometimes David Botstein has been referred to as one of the originators of modern human genetics, in that he was instrumental in the discovery of what was called RIFLIPs, restriction fragment length polymorphisms, that ultimately gave rise to some unique methods of human gene mapping. Tim also did his doctoral work in the laboratory of Phillips Robbins, Phillips Robbins was a MIT professor, and he was considered a great figure in glycosylation, especially in glycosidic lineages. As we move up the ladder, we are related to H.E. Carter, who looks so dapper in this picture. And he is from the University of University of Illinois. And for most of his career, he was a biochemist who was an expert in sphingolipids. Myron Levine is also shown here, as David Botstein did his postdoctoral work with Myron Levine, and he was a professor at the University of Michigan who recently died, but was one of the greats in genetics, especially focused on the genetic regulation of lysogeny and the bacteriophage P22. The lineage through Botstein goes through Tracy Sonneborn. Tracy Sonneborn spent most of his career also at the University of Illinois, where he worked on inheritance in the paramecium. Okay, so as we move forward, or backward actually, what we see is the lineage goes through a guy named Carl Shipp Marvel. I think it is appropriate that Carl Shipp Marvel is in our lineage for Sharon because he was nicknamed Speedy Marvel. He developed polybenzimidazoles, which are temperature-resistant polymers that for the aerospace industry that were intended to take the place of asbestos. He derived from William A. Noyes, who was a pioneering chemist at the University of Illinois, and he was a pioneer in determination of atomic weights. So back in the late 1800s and early 1900s. He derived from Ira Remsen, who was at Johns Hopkins and discovered the sweetener saccharin. Remsen came from the laboratory of William F. R. Fittig, and I thought that it was very interesting that Wilhelm Fittig was a German scientist who discovered the Fittig reaction for synthesis of urea. And altogether, I appear to have lost a Wooler in my notes. But in any case, we'll skip Wooler from now. I know he was a synthetic chemist. So we have a long lineage of synthetic chemists that derive from a guy named Jöns Jacob Berzelius at the Karolinska Institute. And some have referred to Berzelius as one of the fathers of modern chemistry. And indeed, he's a Swedish chemist, and his accomplishments are celebrated each year in Sweden, so you'll have to remember this, Sharon, on August 20th on Berzelius Day. Okay, so that's your family from the academic point of view. And so I think I would be remiss if I didn't actually offer some advice, and I think that we could all offer Sharon some advice from this particular picture. But I think that it is always an amazing time to consider the different passages of our lives, the way we navigate waters. And so if I had any advice, I think that it would be wonderful if you can keep steering the boat with just your foot, but you might actually have to use two hands sometimes. And so with that, I'd like to just wish you all the best, Sharon.
All right. I'd like to thank you all for being here. I'm so excited to see so many people that I know and care about, and I'm really excited to tell you about my thesis work that I've been working on for the last 5 years on characterizing human cellular reprogramming and X chromosome inactivation through single-cell gene expression analysis. So I just want to start by talking a little bit about reprogramming and induced pluripotent stem cells, because I really think that they are the future of regenerative medicine. Now, reprogramming is a process that converts fibroblasts, or skin cells, like everyone in this room has, into induced pluripotent stem cells. And this was discovered in 2006 by Shinya Yamanaka. And just by adding 4 transcription factors to these cells, you can convert them into iPSCs, which are similar to embryonic stem cells. Now, the great thing about iPSCs is that they can self-renew, so that they replicate themselves indefinitely. So you can make as many of these as you would like. And when you have as many as you need, you can then differentiate them into cells you might want. So maybe you're suffering from a neurodegenerative disease and you would like brain cells or neurons. Maybe you've just had a heart attack and you'd like to replace those cells with heart cells. And perhaps you have muscular dystrophy and you would like to make new muscle cells to replace those damaged and diseased cells. So as I'm sure you're all aware of, humans come in 2 types. So you can actually do this, you'd want to do this in both males and females to make sure that both men and women can benefit from these potential therapies. However, over the last 10 years, a lot of work in induced pluripotent stem cells has focused mainly in male fibroblasts to male stem cells. And in some cases, which I'll talk about later, you can't even convert female fibroblasts into stem cells. And so why might that be? What are the differences here that might cause this? So males and females differ at the X chromosome. So all humans have 23 pairs of autosomes, represented here, 1 through 23. And they differ at the sex chromosomes. Males have 1 X chromosome and 1 Y chromosome, and females have 2 X chromosomes. Now the Y chromosome sets up male-specific traits. And that leaves us with an unequal expression of the X chromosome. Women have twice as much, and males just have 1. And so mammals have evolved a really interesting biology that actually silences one of these X chromosomes, and that's known as X chromosome inactivation. So I want to walk you through what we know about X chromosome inactivation in early human development. So all the women in this room started out as an egg carrying 1 X chromosome from their mother and a sperm carrying 1 X chromosome from their father. At fertilization, you have both a maternal X chromosome and a paternal X chromosome. Now this embryo will divide, and each cell that results from this division will end up with both X chromosomes. But at some point during development, likely in one of the pictures that Renee showed between day 2 to day 3 of human development, 1 X chromosome is randomly turned off. And I'm illustrating this with these sort of black hash marks here, and they actually represent exist. Exist is a non-coding RNA that actually physically coats the entire X chromosome and effectively turns it off. And because of this, women then have 1 effective copy of the X chromosome. Now what's interesting is that this process is totally random. Each cell is making its own decision, and so it might turn off, in this case, the paternal X chromosome, leaving only maternal transcripts expressed. However, the neighboring cell might decide to turn off the other X chromosome, leaving only paternal transcripts. And so what's really interesting is in females you have mosaic expression, meaning that not all cells in the body are genetically identical. They're not expressing the same genes necessarily. So I'd really like to think of this process, and in fact my whole PhD was based around this. How do we think about X chromosome inactivation in the process of reprogramming? So we know we're starting with a fibroblast that has decided to inactivate 1 X chromosome or the other, and then we're reprogramming it back to an induced pluripotent stem cell, which is supposed to replicate an embryonic state, a state that hasn't yet adopted a cell fate. So, like those pictures I showed of the developing embryo, do we have 2 active X chromosomes? What's going on here? So, as I am sure lots of you know, many people have actually studied this. It's a huge area of research. And it turns out that female iPSCs are really heterogeneous at the X chromosome. So what I'm showing you here is a picture of a nucleus. It's actually a picture of an induced pluripotent stem cell nucleus. And what's shown here in green are actually markers of the X chromosome. And the fact that we see 2 says that we have 2 active X chromosomes in this iPSC. Now this would suggest that we've reactivated the X chromosome and we've gone back to a state of 2 active X chromosomes, denoted XA, XA. However, a lot of other research has shown that you can find human-induced pluripotent stem cells that are XA, XI, or that they have an inactive X chromosome. And so what I'm showing in this picture is a field of cells, again, where blue is the nucleus, and everywhere you see a red dot, we're actually visualizing exist, that non-coding RNA that's coding the X chromosome. So where you see a red dot, we're actually visualizing the inactive X chromosome. And you can see that most of the cells in this spread have an inactive X chromosome. Even more research has come up with another X chromosome state, and that's known as XAXE, or eroded X. And this refers to the fact that over time you see a loss of exist as you keep cells in culture over time. And so, why do we care? So what? There's 3 states. Exist is on, it's off, it's changing. Why does it matter? Well, 2 years ago, a paper came out that showed if you lose exist expression, mice will get very, very aggressive cancer and die. And so what I'm showing here is a survival plot. We're looking at the number of mice that are alive over a particular number of days. And these groups of mice all have exist, or they're male, so they don't care about exist. And these 2 groups of mice, they actually don't have exist expression, and therefore are unable to inactivate their X chromosome. And they die much more rapidly than their wild-type siblings. And what's really fascinating is that they get really, 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 really bad cancer. So what I'm showing here is the liver, kidney, and lymph nodes taken from these mice. And I'm showing a wild-type mouse, which you could think of as one of these mice, and then 2 mice who are from these groups. And you can see that the liver and the kidney they develop tons of tumors, and that the lymph node is incredibly enlarged in these mice. And so if we think about this in the context of reprogramming, we can make male fibroblasts into iPSCs, and we can make all these awesome cells. But in females, if we don't know what we're doing with the X chromosome, we might end up reprogramming to iPSCs and ending up making cells that are more prone to cancer in women. And so for my PhD, I decided not to worry about male cells, but really to focus on the conversion of female fibroblasts into induced pluripotent stem cells, focusing on understanding X chromosome inactivation to try to understand what these different states mean and what are the consequences for regenerative medicine. Now, there's one thing I didn't tell you about when I showed you these pictures. So this was all literature that came out early in my PhD that I knew about. And when I was looking into this work, one thing that I noticed is the passage number that these cells were assayed at. So first I just want to introduce passage number very quickly. So I told you that induced pluripotent stem cells can grow and divide indefinitely. However, you can imagine that if you let them grow and divide indefinitely, they'd grow out of whatever they were living in and cause a huge mess. So what we do in order to keep these cells in culture over long periods of time is we passage them. And that's what P stands for here. So every time you take a small group of cells and you move it into a slightly bigger dish so they can continue to grow and be happy. And so every time you do that, you increase the passage number. So passage number is also sort of a measure of how long cells have been in culture. So cells at passage 1 or passage 2 haven't been in culture very long. Cells at passage 100 have probably been in culture for years. And so what's really interesting about this work is that these cells were actually they were assayed between passage 3 and passage 15. So these cells could have been in culture for months before people actually looked at what was happening at the X chromosome. And so it seemed to me that if we know what's going on in a somatic fibroblast here, we know it has an inactive X chromosome, and we know that we reprogram and we end up with 2 different states at the other side, 2 active X chromosomes, 1 inactive, 1 active. But we're looking way out here at passage 15. So it seemed to suggest to me that we had to reach a state where we have 2 active X chromosomes, and then perhaps over time, even as short as those 3 passages, we either inactivate the X chromosome before the cells were even looked at, or you're able to maintain this state until those cells were looked at. So I focused my PhD on trying to understand what's happening immediately after reprogramming, as early as passage 0, before cells have been in culture for any period of time. And so, before we can even look at what's happening at passage 0, we actually have to be able to make female iPSCs. And in our lab, we were really excited about using mRNA reprogramming as a way to reprogram cells. And that's in contrast to viral reprogramming that has been used since its discovery in 2006. The exciting thing about mRNA reprogramming is that it doesn't require the integration of the reprogramming factors into the genome, which is useful if you want to then use these cells in future clinical therapies. So this is the protocol we use. We plate our cells at day 0, and we transfect every day for 11 days. Now I understand that not all of you are mRNA biologists, so I just want to let you know what this sort of looks like. So here we have our cell and our nucleus, and out here we have our mRNA for our reprogramming factors that I mentioned before. Every single day we transfect those mRNAs into the cell. They don't actually go into the nucleus, and they're not integrating into the genome. Once they're in the cytoplasm, they're bound by ribosomes, they're translated into proteins, in this case I'm depicting SOX2, and then they move into the nucleus where they're actually able to epigenetically change these cells and convert them from fibroblasts into iPSCs. So after 11 days, you should be able to see this change. And so what I did is I used both male and female fibroblasts to reprogram. And what I'm showing here is day 7 through day 16 of the reprogramming process. And so what I hope you can see in the male line is that on day 12, we start to see a very little colony coming up. And at least by the contrast, you should be able to see that something here on day 16 is growing. So we're able to get really robust colonies by day 16 using this protocol. However, despite many, many attempts of using this method in female cells, I was never able to get female iPSCs using this method. But it was about this time that I was struggling through trying to figure out how am I going to study X chromosome inactivation if I can't even make iPSCs to begin with, that some literature came out that said that microRNAs could increase the efficiency of reprogramming when added to a cocktail of traditional reprogramming factors. And so I troubleshooted a protocol that adds microRNAs on the 1st and 5th day of the reprogramming process, and voila, it was just as easy as that. You can see that when you add microRNAs to female fibroblasts, and again, note that the timeline is actually changed. We're starting at day 1 to day 9. On day 7, and this actually happened every single time we reprogrammed using this method, on day 7 you could see colonies coming up, and by day 9 we had really nice robust colonies forming, and these colonies we could pick at day 11 and passage indefinitely. So the first thing we did when we got these, because at this time no one has actually used mRNA reprogramming and then characterized X chromosome inactivation, we were really interested in what was going on in the X chromosome. So I'm showing you here a similar picture to what I showed before, where we're looking at the nuclei of female iPSCs at passage 5. And I'm showing you here exists this non-coding RNA that's silencing the X chromosome. And so what I hope you can appreciate is almost every single cell in this picture has a mark of exist and has an inactive X chromosome. And that's quantified over there on the right-hand side. Over 90% of cells have an inactive X chromosome. And so this is great, but again, we really wanted to go back and see what's happening immediately after reprogramming. Who knows what could have happened in 5 passages as we're growing these cells? So I developed a protocol that takes fibroblasts on day 0. We reprogram them using mRNA, which I showed you before. And then once we have newly reprogrammed iPSCs on day 11, we do 2 things. So the first thing we do is we manually pick and expand to start passaging some of these iPSCs. However, with all the remaining cells that are in the dish, we trypsinize them into single cells, and we use fluorescence-activated cell sorting to get only the cells we're interested in, only the iPSCs. Now, in order to do this, we have to stain our cells with 2 stem cell markers, TRA160 and SSEA4. And because we're looking for cells that are truly pluripotent, we're pulling out just the double positive group of cells. Once we pull out those positive cells that we are confident actually are our stem cells, we're able to sort them and look at 4 genes in these cells. So I just want to walk you through what the data look like before I explain to you what's going on. So the first thing, as I mentioned, we're looking at 4 genes. The first is exist, which you guys should all be familiar with now. Wherever we see exist expression, we know that there's an inactive X chromosome. Then I have 2 pluripotency markers where these genes are expressed. We know we're looking at stem cells and not the fibroblasts they came from. And then finally I have a housekeeping gene here, just so that we're confident that the cell we're isolating is in fact healthy and doing what it should be doing. So on these plots, what I'm showing is gene expression, where each column on here represents the expression of a gene in a single cell. And each single cell is represented 4 times because we're looking at 4 different genes. So it'll be represented here, 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 and here. So the first thing I hope you can realize is that when we look at our iPSCs, is that they have our pluripotency markers here in blue and in green. And interestingly, we're looking at our passage 0 cells. So these cells have never been passage. They just spent those 11 days reprogramming, and that's it. And we were really surprised to see that they were actually characterized by X chromosome inactivation. Almost all of our cells had exist. And so I mentioned that we also took some cells out of those dishes to grow for a period of time. So we also looked at those cells over time. Oh, yes. So passage 0 has an inactive X chromosome. So we looked at early passage, at passage 3. And what I hope you can appreciate is that we start to see cells that are losing exist expression. They no longer express exist. And I do want to mention just quickly that there are 2 groups of cells here. Cells that were grown in 5% oxygen, which is physiological. It's the oxygen that cells in your body experience. And 20% oxygen. That's atmospheric oxygen. If some cells were in a dish in the air right now, they'd be experiencing 20% oxygen. And so after passage 3, we continued growing and growing them out actually for quite a long time to passage 20. And you can see a really significant drop in the number of cells that are actually expressing exist, suggesting that these cells are becoming XA, XA. Now there's one obvious exception to this rule, which is these group of cells down here. They are an adult cell line and they were grown in 20% oxygen. And it suggests that perhaps some interaction of this cell line in this oxygen tension is actually able to maintain X chromosome inactivation better than others. I like to look at this another way because I think it's a little bit simpler to visualize. What we're looking at here is the percentage of cells that are exist positive over time. So we start with fibroblasts that we know have exist. They have an inactive X chromosome. But then over time, here's passage 3 and here's passage 20, we're seeing a loss of exist expression. And again, it's not as obvious in the adult cell line because we do see this one condition where we don't lose exist as quickly. However, this suggested to us that maybe just being in culture, if you just keep cells in culture for a long period of time, maybe they lose exist. Maybe this would happen to muscle cells. Maybe this would happen to heart cells if you just kept them around a long time. And so we wanted to look at just the fibroblasts. So I'm showing you a picture that's almost identical to the one I showed you before, except for you'll notice that we're not reprogramming, we're just continually culturing fibroblasts. And we're not pulling out a double positive population because we only want fibroblasts. So we're pulling out a double negative population, and we're looking at these same 4 genes in fibroblasts. And so one of the most important first observations is that our fibroblasts don't express our pluripotency genes, so we know that they aren't stem cells. And we also see that they're characterized by exist expression and therefore have an inactive X chromosome. And we can continue to culture them in 5% or 20% oxygen, and we don't really see a change in the percent of cells that are expressing exist. And even if we culture them out to late passage, they still are able to maintain exist expression and an inactive X chromosome. And so again, just to look at it in terms of the percentage of cells that are expressing exist, we don't see a significant change over time, really suggesting that the loss of exist is something that's specific to induced pluripotent stem cells and specific to pluripotent biology. And so just to sort of summarize this, we can sort of see changes over time in terms of what's happening to the X chromosome. And on top of that, there appears to be some effect of what's happening in different oxygen tensions. Now, this raised a really interesting question. Just because we're losing exist, does it mean that these cells are otherwise equivalent? Maybe we're just losing one thing, but maybe everything else is totally fine. And so, because we used single-cell biology, we were actually able to answer this question really, really well. And the reason we want to use single-cell analysis is because it can detect subpopulations of cells. So let's say we have two groups of cells, scenario 1 and scenario 2. And both these populations of cells are made up of exist positive and exist negative cells, just like our iPSCs that we were looking at. Now, in the first scenario, maybe we're looking at a gene that is restricted to the exist positive population. So only cells that have exist are expressing this gene. Well, in scenario 2, all of the cells in our population are expressing this gene. Now, if we took all of these cells and looked at gene expression, and all of these cells and looked at gene expression, they would average out to be exactly the same, and you would miss this underlying biology that this gene is associated with exist positive cells. And so because we're able to look at individual cells, we're able to say what cells that are exist positive have a given gene expression, and what cells that are exist negative have a given gene expression. And so in order to look at whether these cells are equivalent, is we looked at genes on the X chromosome. And there are two types of genes on the X chromosome, and they are escape genes and silence genes. Now, escape genes don't care what's happening to the X chromosome. They don't care if it's inactivated. They're always expressed. So you get one copy from each chromosome, whether exist is on or whether exist is off. And that's contrasted to silenced genes. And silenced genes obey the rules of X chromosome inactivation. So if the X chromosome is turned off, they are not expressed. So when exist is on, you only get one copy, and when exist is off, you get two copies. So if we looked at the expression of this gene in two different populations, you might expect something like this. For an escape gene, the populations should look identical. And what I'm showing here is a density histogram of the normalized expression value. And the mean value of cells shouldn't change. We always get the same expression whether exist is on or exist is off. So the density should look the same. However, for silence genes, when exist is off, you should expect an increase in expression of that gene so that the median expression value is actually increased. So we wanted to look at X chromosome genes in our cells that were exist positive and exist negative to see if we're seeing the shift and if we're seeing sort of a reactivation event of the X chromosome. Okay, and so the first one we looked at was an escape gene. And you can see in the fetal cell line and in the adult line, although this difference is slightly statistically significant, you're not seeing a difference in the density plot and the median expression value of this gene. However, when we look at our silence genes, and especially I'd like you to focus on the adult one, we see that the exist negative population in black is actually statistically significantly increased compared to the exist positive, suggesting we're getting more gene expression off this potentially reactivated X chromosome. However, it's a little bit problematic when you then look at the fetal cell line, because while there is a statistically significant difference, the relationship is opposite. Our exist negative cells, which should have higher expression, actually have less expression. And so this was a little bit puzzling, and what we could say is that we know these states aren't equivalent because we can see differences in gene expression based on whether exist is on or off. But it doesn't necessarily mean that the X chromosome has been entirely turned back on. It seems to suggest that X chromosome expression is really dynamic when you start to lose exist, and that there are some changes that are going on the X chromosome that aren't necessarily happening across the entire X chromosome equivalently. But because we are able to see these differences in these cells, we really wanted to know more about what other differences. We looked at the X chromosome, we looked at these few genes. Are there other differences between these cells over time? And so again, I'm going to bring up this exact same picture where we take fibroblasts and we're able to reprogram them. But instead of just looking at 4 genes or a few genes of interest, we're actually able to look at 53 different genes in each one of these single cells to try to get a better picture of what on a, I was going to say global, but anyone who's a computational biologist or genomics person, that's not global. Taking a slightly larger picture of what's going on in these individual cells. And so I just want to walk you through exactly how we look at 53 different genes in one single cell. So we start out by fact sorting single cells into a single tube, and we add primer pairs for all of the 53 genes we're interested in. Now it's important to note that you have to know what genes you're interested in beforehand, because you have to design primers that are specific to those genes. We then do a reverse transcription and targeted amplification of just those genes in one tube, and then we remove any unincorporated primers, so we just have amplified cDNA of our genes of interest. And we do this for however many single cells we have. We then load these pre-amplified single cells into a microfluidic chip. And on the other side, we load single primer pairs. So where we have a single pre-amplified cell and a single primer pair meets, we get the amplification of just that gene, and we're able to quantify that. And so then we're able to extract amplification data for all of our cells and all of our genes, normalized, and then look at the data. So before I show you what it looks like, I just want to go over the types of cells we looked at. So I looked at over 1,300 single cells, and I just want to go through some of the descriptive characteristics of it. So I focused on fibroblasts and induced pluripotent stem cells across 5% and 20% oxygen, and also across a number of passage numbers. So passage 0, passage 3, and passage 20 across oxygen levels as well. And then in addition, we had a few controls, both male and female embryonic stem cell controls, and a male fibroblast control. And so if we look at all this data together, we have this gigantic heat map. And a heat map is showing relative gene expression across single cells. Wherever you see blue, there's little or no expression, and wherever you see red, that's our highest expression. Across the columns are single cells, and each row represents an individual gene. Now, I don't expect you to actually be able to read these genes, but I will tell you that they fall into 3 major categories. The first is housekeeping genes, because we want to ensure that our single cells are expressing genes that we know that they should. The second group is pluripotency, because we want to know that our stem cells are indeed stem cells, and these are known markers of pluripotency. And then finally, we focused on the X chromosome. Now, with this heat map, we wanted to first just look at how do our individual cells group together. And so we can label each cell in this heat map by different properties. So the first property we looked at was the major biological difference of these cells, whether they were fibroblasts or whether they're induced pluripotent stem cells. So what I'm showing here in black are all the fibroblasts, and in gray, all the stem cells. I'm using unsupervised hierarchical clustering, which means we didn't tell the program which cells were fibroblasts and which ones weren't. It's actually able to distinguish these two groups based only on their gene expression. The next category we were interested in was oxygen tension, because that's something that we know about all of our cells. And so in blue, I'm showing 20% oxygen, and in green, I'm showing 5% oxygen. And so you can see that the grouping of these cells isn't as good as pluripotency, that they don't, all the blue doesn't group with all the green, but you do see some green cells grouping together, more so than interspersed. And finally, the last characteristic that we were really interested in was passage number. Now, passage number is only relevant for our induced pluripotent stem cells, and I'm sorry for any of you who potentially can't see this, but what we're looking at is we're looking at 3 different passage numbers. We're looking at passage 0 in purple, passage 3 in pink, and passage 20 in orange. And what stood out to us in this is that it seems that passage number groups together much better than oxygen tension does, suggesting that maybe there are some genetic differences between passage 0, 3, and 20 that actually separate these groups of cells. And so we did another sort of separation analysis, which is principal component analysis. Now, principal component analysis takes all 53 of those genes and the variation of those genes and reduces it down to a 2-dimensional space, representing the principal components of variation that are the highest. So what we're looking at here are principal component 1 and principal component 2. Now, this graph represents all 1,300 single cells I looked at, and what we then did after we ran the analysis is we labeled cells based on properties we know. So we labeled cells based on iPSC and fibroblasts, And what you can see is that in general, these two cell populations separate very well, meaning that the first two principal components explain most of the variation we see between fibroblasts and induced pluripotent stem cells. However, as I mentioned before, we see some separation of passage number, so we're really interested in how passage number was potentially also separated using principal components. And so we took just our pluripotent population, so just the iPSCs, and again, we're graphing here the second and the third principal component, and we're looking at passage 0, passage 3, and passage 20. And what was really striking and really interesting to see is that you see very good separation of these three groups. So over here we have passage 0, in the middle we have passage 3, and over here we have passage 20. And this sort of recapitulates what we understand about the sort of temporal order of passage 0, passage 3, and passage 20. And we were really interested to try to look and see if we can separate this really well with principal components. Perhaps there are specific genes that are changing over time that actually predict whether a cell is passage 0, passage 3, or passage 20. So in collaboration with Joe Davis in the Montgomery lab, we tried to predict whether a gene was able to predict pluripotency or not alone. So given the expression value of one gene, could we say a cell was pluripotent or not? And what was really surprising to me is that actually 42 of our 50 genes were statistically significant predictors of pluripotency, meaning given one gene, you could predict whether a cell was pluripotent or not. And so here, this is actually the cutoff. So all of these genes are predictive. And what was a really nice sanity check for us is that most of the known pluripotency genes, shown in red, are the most predictive of pluripotency, which is what you'd expect. But excitingly, there's a gene that wasn't a known pluripotency gene. It's NLGN4X. And it was our highest non-pluripotency gene that was actually predictive of a pluripotent state. And I'm going to come back to that in a little bit. Now, this was great. We had 42 genes that were predictive, but it was actually not a great model because there were so many genes that were involved. And so we wanted to reduce the complexity of this model. And we did so using a statistical method known as lasso. And lasso will actually take two genes that are predictive and correlated and then replace it with only one gene of the highest effect size. So you're removing genes that are giving you effectively some of the same information. And so when we try to predict pluripotency using this method, what we see is we come up with 9 genes that are statistically significantly predictive of pluripotency. And again, we see known pluripotency genes coming up, but what was the most exciting to me is that we see NLGN4X, the gene I mentioned before, being our top predictor of pluripotency. Now I think it's important to actually look at what the data are in single cells, and so what I'm showing you here is just a box plot of expression in single cells. And it's grouped by sort of 5 different categories. So the first category here is fibroblasts. Here is passage 0 iPSCs, passage 3, passage 20, and our human embryonic stem cell control. And so you can see that the expression is almost 0 in fibroblasts. You do have a few fibroblasts that have high expression. But in general, it's very highly expressed, both in early and late passage iPSCs, as well as human embryonic stem cells. And so I think it's really important to contrast this to our lowest and non-significant predictor on our list, which is beta-actin. And you can see that in fibroblasts and in all passages and in our embryonic stem cells, we don't really see a change in expression. So it really does seem that just knowing the expression of this gene, you could actually say whether a cell was an iPSC or a fibroblast just with its expression alone. So we're also interested in trying to predict some of the other variables we know, and the most exciting for me being passage number. And in fact, we were able to find 11 genes that were statistically significant predictors of passage number. The strongest being PRMT5 and ALPL, which are both known pluripotency genes. And again, I just want to show you what this looks like in individual cells. You can see in fibroblasts we have a range of expression values, but the expression of PRMT5 is high in passage 0, sort of intermediate at passage 3, and almost 0 at passage 20. And this is really exciting as a stem cell biologist because PRMT5, and we could debate it of course, but is shown to be important in germ cell development. Now, if you are a germ cell biologist and you are trying to make germ cells from induced pluripotent stem cells, you could imagine that you would want the expression of germ cell genes already turned on. So if you started with a population of cells here that don't have this gene turned on, it may be less efficient for you to convert into germ cells than it would be if you started with a population that already had this gene expressed. And sure, this is just one example, but it suggests that genes may be changing expression over time, and depending what downstream application you want to use these cells for, you may want to understand how your genes of interest are changing over this time to best optimize for your particular downstream application of interest. And again, I just want to contrast this with our lowest hit, which again is low in fibroblasts but doesn't change across passage numbers in iPSC. And finally, just for completeness, we wanted to look at oxygen tension. We're able to come up with a list of 11 genes that are actually statistically predictive of oxygen levels. The trends aren't quite as exciting when we look at individual cells. We do see a difference in 5 and 20%, and we can contrast that with a gene on the bottom of our list, which isn't actually changing across oxygen tension. But this lets us build sort of the beginnings of a model, and again, we're just looking at 53 genes and how they change over a few limited biological time points. But we can now characterize genes that are increasing and decreasing as we reprogram cells from fibroblast iPSCs. We can also start to understand what genes are changing as we move from early iPSCs to late iPSCs, and again, what genes are changing when you grow cells in one culture condition versus another. And I think this really suggests that there's a lot we don't understand of how our culturing conditions are actually changing gene expression, and really shows that there's a lot of work that we could be doing to understand these variables and how they affect gene expression in iPSCs. And so with that, I just want to sort of wrap it all together. I've showed you that X chromosome inactivation is changing over time, and that these cells, even though they're just changing the expression of exist, it's actually changing a lot in the other gene expression in these cells. We know that 5% and 20% oxygen has, that there's some differences in how X chromosome inactivation behaves in these two conditions. We've also shown that in order to reprogram female fibroblasts to iPSCs using mRNA, you have to have the addition of microRNAs. And we also know that NLGN4X, the expression of this gene, is really predictive of whether you're in a pluripotent or a somatic cell state. We also know that PRMT5, the expression of PRMT5, is really predictive of whether you're an early iPSC or a late iPSC. And again, that ALPL is the highest predictor in terms of whether you're grown in 5% or 20% oxygen. And so with that, I'd really like to thank the people that helped with this work. Renee for everything, people in the lab who've been directly involved in the papers and the experiments I've done, especially these 5 people. And I'd also really like to acknowledge Steve Montgomery and especially Joe Davis for the work that he's done in helping me with the statistics and modeling gene expression. I'd like to thank Wendy for all her help over the last 5 years, Patty and Jennifer and the FACS facility for all the FACS that I've done, my thesis committee, my chair, Julie, and my fellowships and training grant, and I would like to take questions at this point. Justin.
I have a couple questions. The first one is, you looked at how the gene expression changes over time.
Yeah.
And one of the main things we're interested in is this excess gene and how that's related to that. But can you compare your data set to like, or has anyone compared the female iPS cells to male iPS cells to see? Because I think I remember Conrad, our first year, Conrad Hoffman was there,
yeah,
like that, talking about how gene expression in cells changed over time in iPS cells. I'm just wondering if a lot of these are common or which ones are different. Like,
so that's a great question, and honestly, I don't know how males change over time specifically. I will say that I think some of it is female specific, especially the X chromosome, and how that's changing over time is female specific. Honestly, I spent a little bit of time trying to look at other people's data, and not a lot of people have consistent replicated data over time where you would necessarily be able to compare. So it's hard to say, actually.
And I was also curious, just you used a ton of different individual cells with this method you had, but are they all from the same iPS cell line, or do you do different IPS?
Yeah, so throughout the work, they started from 2 different female fibroblasts, but there are sort of like both of those female fibroblast lines were used in every single condition. So there are 2 cell lines to compare the 2 cell lines together, and they behaved nearly identical. We didn't see a real effect of cell line. But yeah. Yes?
I may have missed this. Why did you look at oxygen tension?
So when I started and I was really interested in X chromosome inactivation, people have actually noted that X chromosome inactivation can be dependent on oxygen tension. So you can derive human embryonic stem cells at 5%, and they will have 2 active X chromosomes, and then you can switch them to 5%, or sorry, derive them at 5%, switch them to 20%, and they will immediately inactivate. So there's definitely some association with oxygen tension and X chromosome inactivation specifically, and so that's why it sort of percolated all single cells. I grew them in both.
Is there any known biological reason for that shift?
No. Katie.
So I noticed that one of your other hits that was like predictive of being a stem cell versus a fibroblast was GAPDH, and I was assuming that was in there because that was in your category of housekeeping genes, but maybe I'm wrong, it's on the X chromosome. And so were there any other like sort of surprising genes that maybe were as a control that could be, where people often use these housekeeping genes, but maybe they really are changing, maybe there are important changes?
Yeah, so almost all of our housekeeping genes changed over time. And it was really surprising. And so I would say that it's, at least for us, looking back, normalizing to housekeeping genes is a terrible idea. They're changing significantly. We had X chromosome genes that were just, no matter what you did, they stay the same. So it's actually one of the other sort of side things that came out were related to X chromosome biology and looking at female cells over time. There are some X chromosome genes that are much better controls than GAPDH and beta-actin and heat shock proteins, for example. Yes, Stephanie?
Have you or has anyone else actually tried to compare like cells with an active X and cells with an inactive X to see if like, I don't know, one makes muscle better and one makes neurons better or something, or is anyone able to get like a large enough number of those?
Not that I know of, and that would be really, really great. That's something that I tried to do, is to come up with a way to separate exist positive and exist negative cells so that you could then assay those cells. The only way I could do it is by killing them, and so as far as I know, and this is specifically in human biology, right? So mouse biology is a little bit different, and you have genetic tools that might be able to let you label one versus the other. But in humans, no one has actually been able to separate these 2 populations and do functional stuff. I know. I tried for 6 months. Okay, I'll just take a few more questions. Lisa.
So I was just wondering, related to Stephanie's question, if it's known what happens to the X chromosome state in differentiated iPS cells?
Yeah, so again, I think there's data out there that supports a wide range of things. Anecdotally, in our lab, once you lose exist expression, even when you differentiate, you don't get it back. It's gone. And so I think that's why it's really sort of alarming, because you might imagine, oh, well, if you differentiate, so you're in this embryonic state and it's doing whatever, but as soon as you go to a neuron or a heart cell, maybe you get it back, and we don't see that. Okay, last question, Cameron.
Okay, so I just want to clarify one thing. So PRMT was low at passage 0 and high at passage 3?
Sorry, no, it was really high at passage 0. I can go back to that slide.
And then low at passage 3?
It was intermediate at passage 3.
Okay. Either way, the question becomes what's changing, right? So so cell division is obviously one possibility. They're dividing more during those passages. So there's
right,
any other
but
changes
I guess we like to think that passage 0 iPSCs are no different than passage 3, right? If we not thinking of this, that they should be dividing at the same rate, right?
Right, but they've undergone more division.
Right, they've undergone more divisions, right?
So
maybe it's an erosion of epigenetic regulation of this,
right, or if you just stop them from dividing, would they still have the state?
I have no idea. That's a good question. I don't actually know. Okay, I do want to end with some personal acknowledgments. Okay. Actually, let's just go back. I'm not going to bring that up yet. Okay. Everyone.
You don't bring it up again.
please bear with me. I wrote it all down so I don't forget everything, but I do have a lot of people to thank. Okay, so as I was preparing for this talk, I was uncharacteristically anxious. As many of you know, I love talking, and I love being in front of an audience, but something was different this time. I kept saying that I wanted my talk to be perfect. That this was the culmination of everything I've been working on so far, and I wouldn't stand for anything but the best. But again, I began realizing that my fears were not so much about giving the best talk, but rather making sure I adequately thanked people for getting me here. There are so many people who share in this accomplishment with me, and while I can't possibly thank you all, I hope you get a glimpse of how much I deeply appreciate your friendship, your mentoring, your support, and your love over the last 5 years. Okay. I've been waiting since this picture was taken to use it in my acknowledgments, and I think it was 3.5 years ago. Renee, I don't think I can thank you enough for giving me the intellectual freedom to pursue science I was interested in. I certainly didn't appreciate it at the time, but I have realized how unique of an experience I was given, and it has made me the independent scientist I am today. The last 1.5 years have been a challenge, but I think my success is due to the fact that you trusted me, and I am so grateful. I also need to thank the Reijo Pera lab, and I tried to capture everyone who's been in lab with me over the last 5 years. Even though we are spread out all over the place, I am so thankful to have spent 3.5 years learning from you. You made coming into lab incredibly fun, and I value all the friendships, beer lunches, March Madnesses, and retreats that we shared. Thank you for your support and teaching me literally everything I know. I also want to thank my second lab, the Baker lab. Julie, you went above and beyond the call of duty to give me a lab home for the last 1.5 years, and I appreciate everything you've done for me. And to everyone in the lab, I want to thank you for welcoming me in and making me feel like I was a part of the group. I need to also thank the people who got me here. I know without a doubt that I'm standing here today because of the incredible people who believed in me before I knew to believe in myself. Bill Brown, who is unfortunately no longer with us, Amy Brooker, and Aaron Mitchell. I'm not sure what you saw in me almost a decade ago, but without your encouragement, I couldn't have been capable of achieving this. There have also been so many groups of people that I have had the pleasure to meet, and you have been a part of some of the most enriching parts of my grad school experience. From soccer to softball, from SSRP to the FemGen minor, I've had a chance to meet incredibly smart, talented people, and I'm so thankful that our paths crossed. I also need to thank the RAIN CAs over the past 4 years for being an amazingly fun group of friends. We've made lasting friendships that I'm confident will last a lifetime. I'm also so incredibly lucky to have the support of friends from my time before I got to Stanford. While many of them don't understand what I do, they have been unwavering in their support and very vocal with their praise of what I've finally accomplished today. Although I don't get to see many of you with regularity, know that I think of you often and value our friendships tremendously. There have been, when I came to Stanford, I had no idea I would meet the most wonderful group of people. You're some of the smartest people I've ever met, yet somehow know that there's more to life than school and research and studying. I've enjoyed every second we've spent together, especially those that include concerts and beer Olympics and road trips and football games and dinner circles and scavenger hunts and birthdays and all the general shenanigans that we've gotten into. You have made these 5 years the best years of my life. The last group of people I have to thank are my classmates. They're honestly not even words I can find to describe how important you've been to me. You inspire me and you challenge me to be better. I'm impressed by your accomplishments every day, and I know we are the most incredible class to pass through this department. No one has played more games of volleyball together or pulled off a better retreat entertainment or has remained as close as we have. I'm not sure I could have accomplished this today without the supportive environment we've created. I'd also like to quickly thank a few individuals. Katie, I'm not sure how I can compute the value of our friendship on my time in grad school. Our daily, or should I say, 3.5 times per week, lunches provided not only escape from lab, but generated truly deep and meaningful friendship. One of my biggest concerns of leaving grad school is not having someone to have lunch with every day. By many accounts, Zach and I probably shouldn't have ended up friends due to a series of unfortunate events at our first football game. But somehow, one fateful Valentine's Day, I was able to convince you to skip school and get drunk with me. I don't know what happened, but it started a friendship that I can't imagine living without. You have been there for me through some of the most difficult parts of grad school and Beer Olympics. And I cherish our friendship more than you know. The last individual I want to thank is Helio. Helio, you once mentioned you weren't sure how we were even friends because we were so different. Maybe it's those differences that make us such good friends. You have never failed to make me feel important and smart and deserving of all I've achieved. I can always count on you to be on my side and there for me whenever and for whatever I need. I'm going to miss our Pachi's dinners, even though you won't. Our walks around the med school and your constant insults that I know are only in good fun. I'm especially going to miss your awkward Nick walk. And last, but by far the most important, I'd like to thank my family. Kat and Andrew, I want you to know how proud I am to be your sister, and how thankful I am that you're here today. I also want to thank my Uncle Howard and Myra for coming in from England to be here. Finally, Mom and Dad, thank you for being my biggest cheerleaders. Thank you for being there to celebrate my biggest achievements, and also there to pick me up from my lowest lows. I wouldn't be here without you, and I love you. Yesterday.
Thank you, Sharon. I think you're making people cry along. I don't know that it could be any more heartfelt or beautiful. And I believe now we go into a private section. Yeah.
If it's raining for the party, just go to Allway Courtyard and we'll just move to the side and text me and I'll tell you where we're going to be.